Hi, my name's Erin and I'm a genetic counselor with SDX Labs. In this video, we will review some basic genetic concepts. We will begin by discussing cells, chromosomes, and DNA. Then we will discuss the two main types of genetic conditions, chromosome disorders and single gene disorders, including autosomal dominant and autosomal recessive inheritance. All animals, including humans, are made up of billions and billions of cells. Cells provide the body with structure, make energy, and perform many different specialized functions. Inside each cell are structures called chromosomes, which are tightly wound packages of DNA. DNA provides our body with instructions on how to grow and develop. Most cells contain 46 chromosomes, or 23 pairs of chromosomes. One in each pair is inherited from a person's mother, and the other is inherited from their father. The first 22 pairs of chromosomes are called autosomes and are the same in males and females. The 23rd pair of chromosomes are called the sex chromosomes, and these determine if a person develops as male or female. Females typically have two X chromosomes, and males typically have one X and one Y chromosome. If we unwind a chromosome, we will find our DNA, comprised of two strands twisted around each other. DNA is written using four letters, or bases, A, T, G, and C. Just like letters in the alphabet, these bases can be arranged in different patterns to communicate how our bodies should function. Now that we've reviewed a few basic genetic concepts, let's talk about the two main types of genetic conditions, chromosome disorders and single gene disorders. Chromosome disorders occur when a person has an extra or missing chromosome, or an extra or missing piece of a chromosome. Because our chromosomes are so important for normal growth and development, many pregnancies with chromosome disorders miscarry in the first trimester. Chromosome disorders are typically sporadic and do not run in families. If a person is born with a chromosome disorder, they may experience intellectual disabilities and/or birth defects. But the possible implications of a chromosome disorder are extremely variable and depend on the size and content of that extra or missing DNA. A common example of a chromosome disorder is Down syndrome, also known as trisomy 21. Down syndrome occurs when a person has 3 copies of chromosome 21 instead of the typical 2 copies. Non-invasive prenatal screening, or NIPS, is a type of blood test that can be done in pregnancy to see if there's an increased risk for certain chromosome disorders. To learn more, please see our Introduction to NIPS video. Now that we've discussed chromosome disorders, let's talk about single gene disorders. Single gene disorders occur when there's a spelling error or typo in a person's DNA. The word gene is used to describe a section of DNA that has a specific job, like a recipe in a cookbook. If the wrong DNA letter is used, the gene may no longer function properly. These typos are called mutations. Because we inherit our DNA from our parents, single gene disorders can run in families. It's important to remember that because we have 2 copies of each chromosome, we also have 2 copies of each gene. Some single gene disorders occur when just 1 copy of the gene is mutated, and others only occur when both copies of the gene are mutated. Autosomal dominant single gene disorders occur when just 1 copy of the gene is mutated. If a parent has an autosomal dominant condition, there's a 50% chance they will pass it on in each pregnancy. Some autosomal dominant conditions you may have heard of include Huntington's disease, Marfan syndrome, or achondroplasia. Autosomal recessive single gene disorders only occur if both copies of a gene are mutated. If a person has just 1 gene mutated for an autosomal recessive condition, they are called a carrier, and carriers typically have no signs or symptoms of the disease. Most people are carriers of a few different autosomal recessive conditions. If a person and their reproductive partner are carriers of the same autosomal recessive condition, each time they have a pregnancy, there's a 25% chance that the baby will have that condition. Some common examples of autosomal recessive conditions include cystic fibrosis, sickle cell anemia, and Tay-Sachs disease. Carrier screening is a type of genetic testing that's offered to reproductive couples who are pregnant or hoping to become pregnant to learn if they are carriers of any of the same autosomal recessive conditions. If you are pregnant or hoping to become pregnant and have concerns about any genetic conditions, especially those that may run in your family, you may consider meeting with a prenatal genetic counselor. To find a genetic counselor in your area, visit www.findageneticcounselor.com. To make the most out of your genetic counseling session, it's recommended that you have a detailed family history and, if possible, have copies of any of your family members' genetic test results. At SDX Labs, we believe in providing patients with the most proactive screening tools available so that they can make informed health decisions. For more information on genetic conditions or the services offered at SDX Labs, please see the rest of our video library or reach out to us directly.